Hello, good morning, everyone. Thank you for joining us to this webinar in which we will be discussing new applications leveraging the smarter technology for next generation sequencing. So if you have any question during the webinar, please type it in the chat box and we will be taking them at the end of the presentation. So the agenda for today is the following. So first, we will be giving an overview of the smarter NGS technology. And then we will be focusing in three specific applications. First will be targeted RNA-seq, then small RNA-seq, and we will finish by our solutions for immune profiling, comprising TCR and BCR profiling. So let's get started talking about what is smart technology, so we can understand a bit better this technology that you will see incorporated in all of our products. So just let me, SMART actually is an acronym for switching mechanism at the 5' end of RNA template. And here is how it works. So we will be starting the reverse transcription using oligo-DT primer or a random primer, using a reverse transcriptase which has two properties, terminal transferase activity and template switching activity. So when this reverse transcriptase reaches the 5' end of the transcript, it will add some non-template nucleotides represented here as X's. And we do this reaction in the presence of an oligo called the smarter template switching oligo, which contains nucleotides that will anneal to these non-template nucleotides. So after this annealing, the reverse transcriptase can then switch the template. So this is the template switching activity. And it will use this smart oligo as new template, and it will allow it to incorporate this sequence here shown in green at the 5' end. So as a result, we have here first-strand cDNA, which contains known sequences at both ends, and these sequences can be then used as priming sites for photo amplification of the cDNA by PCR. So the advantage of this technology is that it's exceptionally sensitive and also very reproducible. And this is what allows smarter technology to be incorporated in applications that are very challenging. So specifically, we have this technology for single-cell RNA sequencing. And we have also extended this technology for other applications like using degraded samples or prokaryotic RNA. So you can do whole transcriptome analysis using smarter technology, but also we can do some more targeted applications. And this is what we will be discussing today. If you are interested in this whole transcriptome applications, we will see at the end of the webinar where you can get more information. So coming back to today's topic, we will be discussing these three targeted sequencing applications. So let's get started with the targeted RNA-seq. So the first thing that we may ask is why someone would need to do targeted RNA-seq instead of doing the whole transcriptome analysis. So it depends on what are you studying or what is you're looking for. Because if you see here in this graph, most of the reads in a standard RNA-seq experiment actually will correspond to a minority of the RNAs, which are very abundant, while a minority of the reads will correspond to the most of the RNAs, which are actually less expressed. So if you're interested in some of these RNAs which have a lower level of expression, or some specific isoforms or rare events like gene fusions, it may be difficult to identify them, or you will need a very deep sequencing to get them. So in those cases, it's very useful to do targeted RNA-seq to reduce the complexity of the sequencing and improve the coverage of the genes of interest, and also get the information of what is relevant, what is interesting to you. So for that, our solution is the Smarter Targeted RNA Capture Kit. This is a kit that will allow you to generate a library from selected targets using customer-designed probes, and you will be able to target up to 100 genes. So the particularity of this kit is that it targets RNA and not cDNA. It means that you will be, with the specific probes, we will be pulling down the RNA and then doing your reverse transcription only from this specific RNA. So you have here the workflow. You will incubate the RNA with some specific probes. So these are only biotinylated DNA probes. And the design is quite simple. You need only one per exon. And then you will do hybridization, which is quite fast, and no more than 3 hours. And you will have then washing steps to remove this probe. Then it comes the capture. So it's done using streptavidin bits, which are provided with the kit. And these streptavidin kits have been specifically selected because they allow to do the cDNA synthesis. And so you will have your reverse transcription only in your pull down specific transcripts. These bits will be and probes will be displayed during RT. So at the end, you have a cDNA only from your specific transcripts, and you can prepare a library out of it. So here for the reverse transcription, we are using SmartSeq V4 technology, which is our most performant technology for sequencing, and this is what actually we use in our single cell kit. So here's just to show some results for this kit. In this experiment, we targeted some a few genes, 9 genes, which correspond to 160 exons and cover a length of 55 kb, more or less. We analyze here then the fold enrichment when we use our probes. So you can see here that the enrichment is variable. It depends on, for some targets is quite high, 300 times, for other is a bit less, 6 times. But you can also see here that when you are not enriching, like in this case of 2 very abundant genes, GAPDH or this ERCC that we're expecting, there is no enrichment. You can also see here how the kit improves the detection of the genes of interest, because if you do no capture at all, you will be identifying these genes in less than 1% of the reads. While with the capture, the percentage of reads on target varies to from 14 to 37%, depending on the sample. Here, these are the data for 2 different cell lines and for 2 different RNA. We have also evaluated the utility of this approach to detecting some rare events like gene fusions. So for this experiment, we spiked in RNA from K562 cells, which actually they carry a fusion between BCR and ABL1 gene. So this was spiked into human brain RNA at different percentages, and we did a capture with probes targeting only ABL1, so only one of the partners of the fusion. So what we see here is that the targeted RNA-seq approach allows us to identify reads with frequency when this RNA corresponding to the fusion is present at less than 0.5% of the total RNA, showing the sensitivity of the approach. And knowing also that we only use the capture probes for one of the partners of the fusion, so this can be improved if we, for example, include probes to target BCR. Okay, so moving on with the presentation, let's talk now about small RNA sequencing. This is another targeted approach. So sometimes you are not interested in the whole transcriptome, you are only focused in small RNAs. This may include microRNAs, pRNAs, small nuclear RNAs, and so on. So everything that is, let's say, lower than 150 nucleotide. So these are quite challenging samples to use on RNA sequencing. Most of time the samples are precious and you have only small quantities, so you need a sensitive method. Another challenge is that for small RNA sequencing, you cannot use random priming because, for example, if you think about the microRNA, it's too small to the 21 nucleotides to use random hexamers. Also, most of protocols that are out there are very long. They require several steps, and also polyacrylamide gel electrophoresis purification, which can be something time consuming. And another problem of current methods is that most of them use ligation at the 5' and 3' ends of the small RNAs, and this introduces bias. So this has been described already in several publications and is represented here. When you prepare a microRNA library, you can see here that although all these more than 900 microRNAs were originally at the same input concentration, after library prep, only 22% of them are in the expected expression level. So it means that some microRNAs are ligated more efficiently than others. So to cope with these challenges, we have developed the smarter small RNA-seq kit for Illumina. So this kit it's able to prepare libraries from RNAs, a big variety of RNAs, so ranging from 15 to 150 base pairs. So this includes all these kind of small RNAs that you can see here. And we are using SMARTed instead of ligation to avoid this bias issue. So the way it works is just we are adding a poly-A tail to the small RNAs, and this will create an anchoring site for oligo-DT priming, and then using the smarter reaction that I already shown, we can incorporate the adapter at the 5' end. And also what is nice in this approach is that in the PCR step, we are already incorporating the Illumina indexes and adapters. So as a result, at the end of the protocol, we have a library which is already ready to sequence. So this protocol is also quite fast and streamlined, so it will take less than 3 hours to get your library. And then you will have to do the library size selection. And for that, we wanted to avoid the page purification method. So we are proposing two methods that are manual, size selection using Ampere bits. So just using different ratios that have been optimized, and we show in the protocol of Ampere bits, you will be able to size select your library. Or there is an automated gel selection protocol using the Blue Pippin instrument. So this, as I said, is designed for small RNA sequencing, but it can also be used for other applications that are generating RNA which is very small size, like RNA immunoprecipitation, chromatin-linked immunoprecipitation, or ribosomal profiling. So here's just to show how with this approach with not using ligation, we have improved on the bias issue. So we can see here that smarter libraries are more diverse, that libraries prepared with adapter ligation, and we're increasing this by at least twofold. The method is, as I said, quite reproducible. This is one of the features of the smarter protocols. So you can see here that the comparison between two replicates, the correlation is quite high, but also when we are comparing across different inputs. So here we have more than 1,000 fold difference, and the reproducibility is very good. And to finish with this small RNA part, I just wanted to show that it has also been used successfully with clinical samples. So many people are interested in analyzing small RNAs that are found in plasma or serum fraction. So we had collaborators with TRI that worked with this technology, and they could get good data from plasma. Even though plasma, the amount which was used was below the specification of the kit, because this kit is designed to work with 1 nanogram at least of total RNA. But from plasma, sometimes you don't have 1 nanogram. In this case, they got only 400 picogram for 200 from 200 microliters of plasma, but still they were be able to identify microRNAs. In this case, this was they were interested in, and also with very good reproducibility, either in the plasma and serum samples or comparing serum versus plasma. So this data is also available on the website if you want to have a deeper look. So now I would like to move to the third application. This is the Smarter for immune profiling, and here we have developed some quite a variety of products that I will be describing. So first we will talk about the products that we have for bulk TCR and BCR profiling, and at the end I will talk about our product for single cell TCR profiling. So before that, let me just briefly talk about, or let's define what are TCR and BCR. So these are receptors that are found in the surface of T cells and B cells. So as you probably may know, T cells have a central role in cell-mediated immunity, while B cells have a central role in the adaptive immune system by secreting antibodies. So essential to these functions of these immune cells are the receptors in their surface. So in case of the T cell, we have on the surface the T cell receptor, or TCR, which is composed of two subunits, TCR alpha and TCR beta. This is in the majority of cells. There are also a minority of cells that are composed of TCR gamma and delta subunits. In the case of B cells, we have the surface, these B cell receptors, which are also called immunoglobulins. And what you can see here is that they are also heterodimers, but they are composed of two heavy chains, represented also as H, and two light chains. And these light chains can be of two types, kappa or lambda, or K and L as shown here. So let's talk first about the TCR, and then we will go back to BCR. So since the role of TCR are to recognize all the diversity of antigens that that an organism may encounter, they require to have high diversity, to have a lot of different combinations that will allow to recognize all these antigens. And this is achieved by some complex mechanisms that are happening during T cell development. So basically, here I'm showing only for TCR beta. This locus contains several segments, which are called V for variable, D for diversity, or J for joining. And during the development of the T cells, there will be some processes called VDJ recombination, in which some of these fragments will be, let's say, cut and paste together. And of course, the combination are multiple. So depending on which V, which D, and which J segment is chosen, you will have a different sequence. Besides that, after this rearrangement, you will have transcription, you will have splicing. So at the end, you will have a specific sequence here, a mRNA sequence, that will give you a protein TCR beta. So in the case of TCR alpha, is a quite similar process, except that TCR alpha locus does not contain the D segment. So the rearrangement will be only between V and J. So the bottom line of this is that in every T cell, different TCR alpha and TCR beta will be present, and this specific region is what most people are interested in characterizing here. This is what is called also a clonotype. So this kit will allow you to identify in all the TCR population what are the different clonotypes that are present there. So before talking about our method, let's talk briefly how people are currently most of the time analyzing this TCR repertoire. So the most common method used so far is multiplex PCR. So for this, you will start from a gDNA, a genomic DNA template, and you will use primers that are sitting into the variable region and the joining region. So as any PCR experiment, and this is particularly true in multiplex PCR, you will have some amplification biases because you will have some sequences that are amplified more efficiently than others. So this requires a lot of optimization. The other thing is that you can only amplify the TCR beta or the TCR alpha in separate experiments every time you need to do separate amplifications. And when you are starting from genomic DNA, you will probably get many more unexpressed or non-functional sequences because, again, after this genomic DNA recombination, you will have splicing, and so you may have some sequences that will not correspond to the actual TCR protein. So the approach that we want to use here is RACE. Why? Because it has some advantages that we will see here. We will be starting from a messenger RNA template, so it means that we will probably have less non-functional sequences as compared to using genomic RNA. Also, we will get the full sequencing data for the whole region. Because when you are using a multiplex PCR, your primers are sitting inside the sequences that you want to analyze. When you use RACE, you can get the full mRNA up to the 5' end. I will show how it works. So you don't need any knowledge of this region. The other advantage is that you are using only a primer per reaction, a primer pair per reaction. So you will have a primer pair for TCR alpha and another for TCR beta. And it means also that you can combine both in the same experiment. So let's see how it looks like in our kits for TCR profiling. So what we are seeing here is the workflow for the smarter human TCR profiling kit, but actually we have the same for a mouse. So it's quite similar, just the mouse will contain mouse sequences. So just to explain briefly how it works, we will start first reverse transcription with an oligo-DT primer. We will be incorporating the 5' end using the template switching reaction. So this is what I said before, that thanks to this template switching reaction, you don't need to have a knowledge of the 5' end. And then it comes the part in which we will be specifically be amplifying TCR sequences. So for that, we do a semi-nested PCR in which we will be using specific TCR alpha or beta primers separately or in the same reaction as you wish. And then in a second PCR round, we will be incorporating Illumina indexes and adapters. So the advantage here is that we will be started with the input RNA or cells, and then we will end with a sequencing ready library. So there's no need for downstream library prep. This is ready to go into the sequencer. As I said, this is compatible with human and mouse. So here's how it looks like the libraries. We put some examples using either fragment analyzer or bioanalyzer. You can see that the picks may look different depending on what you use. And sometimes in case, for example, of JORCAT RNA, which have only specific clonotype, you can clearly differentiate the two picks, TCR alpha and beta. But this is using fragment analyzer with bioanalyzer. Sometimes it's not easy to look like, but these are some examples to show you how it looks like. When we sequence these libraries, we find that this is libraries prepared with PBMC RNA. We can see that most of the reads are on target. They correspond either to TCR alpha and beta in more or less similar proportions as one could expect. And when we analyze the correlation of the the top 100 clonotypes identified in two experiments, one using 100 nanogram and the other 1 microgram, we see that the correlation is very high. So this shows the robustness of our approach. We also evaluate the sensitivity of the approach. So we wanted to know what was the minimum amount that was required to identify a clonotype. So in this case, for this experiment, we took PBMC RNA and we spiked in with JORCAT RNA at different concentrations. And this is because the clonotype of JORCAT RNA is known, so then we can evaluate what was the minimum amount in which we could identify this JORCAT RNA above the background. And what we found here is that these specific JORCAT clonotype sequences could be identified at a concentration as low as 0.1% above the background. So this shows that the SMART approach is able to identify very rare clonotypes. As I said, this kit can also be used with mouse, but it will be actually a different kit. You cannot use a mouse kit for human and vice versa. So the mouse kit comes with the mouse sequences, and also the amplification conditions have been optimized for that. So here we can see data obtained from mouse in which the spleen was taken to isolated splenocytes or CD4 plus T cells. And after RNA extraction, we went to library prep, and then we can see that actually we get good on-target mapping rate with more or less equal proportion of the reads aligning to alpha and beta as expected. And in this experiment, which actually was done by collaborators, they wanted to know which was the depth of sequencing that it was required to identify the most of clonotypes. And what they found is that it depends actually on the sample. So if you take splenocytes, you can see here that you will reach saturation, the number of clonotypes identified, around 300,000 reads, while with CD4 plus cells, which are much more diverse, you need to sequence deeper, so you can go up to 600,000 reads and to reach saturation of the number of clonotypes identified. So of course this can also vary depending on the sample type, and this needs to be evaluated depending on your experiment. So now coming back to the beta cell receptor. So I said before that in the case of beta cells, we will have a heavy chain and a light chain that can be either kappa or lambda. Actually, what happens in the BCR heavy chain and light chain is very similar to what I explained before for TCR. So you will have the same mechanism of recombination, and then you will have high diversity on the sequences that will be shown in these visceral receptors. So what we want in our solution for sequencing BCR is to provide an option to be able to amplify all these variable regions. So this kit is for the moment only available for mouse. It's a smarter mouse BCR IgG HKL profiling kit. So H for heavy, K for kappa, and L from lambda. So you can see that the work for the library prep workflow, it looks quite similar to what I shown before for the TCR. But of course, in this case, we are incorporating primers for BCR amplification. In this case, we will have 3 sets of primers. So one for the heavy chain, and 2 for the light chain. So we will have for amplifying kappa chain or lambda chain. But the workflow is very similar. You can start with your RNA or your cells and then end up with your library ready to sequence. So let's see how it looks like when we obtain the libraries. Here, if we analyze control RNA from spleen, we can see that in spleen you will expect to have a good representation of the kappa and the lambda chains, because you will have a mix of both. This is what we can see here. So we can see a nice peak for the heavy chain and also one for kappa and another for lambda. Here what we are showing is non-template control, and as expected there is no amplification. So these peaks are quite specific. We wanted to analyze hybridoma samples. So here we can see also the difference. So in hybridoma, usually you will have one of the light chains that will be more represented than another. And this is what we are seeing here. So we are able to identify the heavy chain in all the 3 samples. But we can see that in these 3 hybridoma that we analyzed, the kappa chain is the predominant, while the lambda chain is barely detected. So something that is important in this protocol is that we provide the flexibility to the user depending on what you want to study. You can do the RT step only in one tube, and then you can split this to do separately the PCR one for each of the chains that you are interested. And then you can do also the PCR2 separately and pull everything for sequencing. So this is important because maybe, for example, in this case of this hybridome, you may choose not to analyze lambda because maybe you only want to detect the clonotype for kappa because it's the predominant one. So this you can do it. I mean, you can choose to sequence the 3 and at the same time only 2. And what we wanted to see here in this experiment is how this affected the results. And what we see here is, in this case, for example, if we are only analyzing the heavy chain and the kappa chain, so most of the risk will be aligned to immunoglobulin. And we can detect very accurately the clonotype of the heavy chain and the kappa chain. If we include here the lambda library, which as you can see has not much many reads, we are decreasing the percentage of reads on target, but still we are able to identify accurately the CDR3 sequences. And it's the same for this other hybridome. So actually the identification of the clonotype won't be adversely affected. Okay, so to summarize, what are the benefits of our smarter approach to TCR and BCR profiling? The first that you will get the full length VDJ sequence information. So you will have the full sequences for the TCR and BCR mRNA transcripts. You can choose to analyze TCR alpha and beta in the same reaction or separately, and the same for BCR HKL. The sample inputs are very flexible, so you can start from purified RNA, but you can also start from purified T cells or purified B cells. So you will need 50 for human, but you will need 1000 for mouse if starting from cells. The method is very sensitive, as I could show. You will be able to identify low abundance TCR or BCR sequencing. And the other benefit is that you are getting Illumina ready to sequence libraries, and all the indexes are included and will allow you to multiplex up to 96 libraries in the same flow cell lane. So the last point that I want to mention is our latest addition to this immune profiling portfolio. This is the single cell TCR alpha and beta profiling kit. So first, why would be someone interested in doing this single cell TCR sequencing? So as I showed before, with the bulk kit, you will be able to get an information about what are the alpha and beta clonotypes present there. So for example, if you have this bulk population and you get this sequencing data, you will maybe assume that this 5% of TCR alpha and this 5% of TCR beta clonotype will represent a rare population which has this paired alpha and beta clonotype. But if you get this data, 45% for each of these different clonotypes, you don't know actually if this alpha pairs with this beta or is this alpha with the other beta. And sometimes you will need, depending on what is your goal of research, you will need to know precisely for this cell, I have this alpha paired with this beta. So to do so, you will need absolutely to do single cell TCR profiling. So instead of taking the whole population and analyze all the alpha and beta which are there, you will take cell by cell and determine which alpha and which beta has each cell. So for that, we have adapted the protocol that I shown a little bit earlier, and we got a smarter human single cell TCR alpha and beta profiling kit. So it looks similar to the standard workflow that I mentioned before, but there are some key differences. So the main difference is that we are using here in the template switching oligo, we are using an indexed oligo, and we include 8 of them, as you can see here, from A to H. So this allows us to pull samples afterwards. So after the reverse transcription, we will have different indexed cDNA, and also we need to do an additional amplification of the cDNA here because the amount that we get from a single cell is very low. And then once that we have that, we can do the specific TCR amplification, very similar to what it was shown before, and also incorporating Illumina indexes and adapters. So the advantage of using this indexed oligonucleotide is that we can start from a plate having 96 cells, and then we can just pull this in 12 pulls. It means that all this step here, the PCR amplification, will be only done from 12 instead of 96 samples. So it makes it easier to handle and also to analyze. So here is just to show some experiments that we did to see how it looks like with that. So in this experiment, 2 different T cells which known clonotypes were used. So these were the JORCAT again and the CCRF. They were mixed in a 1-to-1 ratio. And then they were sorted by FACS, and we used the smarter NGS protocol. And then we wanted to see if we were able to identify the cells based on the expected clonotype. So this is what we can see here from our 96 plate. We were able to identify the clonotype in 43 cells for JORCAT and 46 for CCRF cells. So this corresponds more or less at the 1-to-1 ratio that we initially put. For 7 cells, we were not able to get enough data to identify the clonotype, so they were excluded. And then we analyze the pairing information. So we can see here that in some of the cells, we are able to identify only the alpha chain. In others, we will identify the beta chain. And in others, we will be able to identify alpha and beta pairing. It's usually quite common, depending on the cell type, to identify or to have a higher expression of the beta change. This is why it is more abundant, let's say, than in the case of the alpha in this experiment. But we can see that still we have around 40% pairing, alpha beta pairing ratio. And what's interesting also to see is when we analyze the bioanalyzer profilings of these pools, we can see that there are some concordances here. So for example, if you take here the pool number 9, in the bioanalyzer, we will see only one peak. So it corresponds to this beta majoritarily expressed. In pool 10, we have most of cells that show the 2 peaks, and this is what also we can see here. So 2 peaks, one for alpha and one for beta. In pool 11, we have a mix of alpha, beta, and beta. So this is quite similar to what is shown. Well, the kit also includes a positive control. This is what we see here. And this is purified spleen RNA. And we don't have any background on the negative control. So to summarize, this single cell TCR approach is very flexible, provides a possibility to work from cells isolated with FACS or manually sorted. You can use the optimized indexing, allowing to pull 96 cells into 12 libraries. It uses the race-based approach that we already discussed that allows for high sensitivity and full-length VDJ sequence identification. And in this case, with it is able to provide accurate pairing information. And important to mention, with this kit, the user will also get access to a demultiplexer software available on our website, and with the license agreement and sample data included to be able to analyze the single cell data. So with that, to learn more, if you want to see more in detail all this data that I mentioned, we have our NGS resource portal here at acarabio.com/NGS. We have webinars for each of these products. We have tech notes and poster with more details on that. So thank you for listening. And now I will check on the questions. So if you have, I see that here there are some questions, and if you have others, please do not hesitate. Okay. So there's a question: what protocol modification are required to use the small RNA-seq kit for ribosomal profiling? Yes. So actually, there is we had an early access customer that used this technology for that, and he published a nice paper, and he provides there all the detailed information. I can share that with you. What is important actually when using this small RNA-seq for that is to do a treatment with a kinase just to restore the 3' OH tail that is required to add the poly-A tail to the small RNA. So this is what he did, and what we recommend when starting with this kind of samples. because as I said, we need a polyadenylation to start with the kit. And we have recommendations for that, so I will send you that along with the paper, which shows the very nice application of the small RNA-seq kit for RiboSeq. Okay, another question is the libraries for the TCR profiling kit are quite large. What sequencers are compatible with? Yes, good question. This I didn't mention. Indeed, here, since you are obtaining the full-length TCR sequence, this depending on the kit, but it will range between, let's say, 600 and 800 base pairs. And for sequencing of this TCR and BCR libraries, we recommend only Illumina MiSeq, and it will require sequencing of 2 times 300 base pairs. This is enough to cover the CDR3 sequence, because you have to know that in this 700 or library size that you see, you also have to take out the size of the Illumina indexes and adapters that are also adding to that. So yes, Illumina MiSeq is the recommendation for this kit. Another one. Just let me check. So can be the small RNA used with plants? Yes, this kit is compatible actually with any kind of RNA, and actually the control that is included in the kit is microRNA derived from Arabidopsis. Do we have specific recommendations for the probe design? So we do say in the user manual how probes should be designed. So actually it's quite simple, because you only need 1 probe per exon, and each probe only needs to be 60 nucleotides. So you will take your gene, if it has 5 exons, you will just define 60 nucleotide region in each exon to do that. And that's it. You don't need to do tiling probes or this kind of things. Actually, we tried this approach using tiling probes, and actually it did not work better than only using 1 probe per exon. So this is what we recommend for the better specificity. But we do not have at the moment any, let's say, automated way of doing this probe design. But if you need help with that, just you can contact us at tech support, and we can also help you with that. Okay. So it seems like there are no other questions. So we will stop the webinar here. If you have other questions, you have here the tech support email address. Please keep sending us your questions. And so thank you very much for listening, and wish you a nice day. Bye.